Hi everyone, welcome and thank you for joining us for today's webinar regarding meeting the challenges of microRNA research. My name is Jonathan Schaefer and I'm a Senior Scientist in Product Development, and I will be your guide for the next hour. Before we continue today, would one or a few of you simply mind sending me a question in the question box saying that you can hear me both loudly and clearly? While we're waiting for audio confirmation, I'll give our standard legal disclaimer. The QIAGEN products shown here are intended for molecular biology applications, and these products are not intended for the diagnosis, prevention, or treatment of a disease. Okay, thanks for the audio confirmation. We'll continue on. Here at QIAGEN, we give a whole host of webinars throughout the month. You can see we're only on the second of the month in March, and we have a whole host throughout the rest of the month. We have a few more microRNA webinars, including the functional analysis of microRNA, as well as plant microRNA profiling. Also, for your interest, we have a few NGS-related webinars nearly every week of the month. So for today's agenda, we want to cover the following. We'll begin with a brief microRNA background, and then spend the bulk of our time discussing your sample prep, real-time PCR assay, data analysis, and data interpretation solutions from QIAGEN. We'll then finish with conclusions, summaries, and a live question and answer session. If you'll note in the WebEx software, there is a handout. It's a microRNA webinar, March 2nd, 2016. And this is a PDF of the slides that I am discussing today. So feel free to download it, mark any slides that you have any questions on, or feel free later on that if you look at it in a week or two from now, feel free to email me any additional questions that you might have. So let's begin. MicroRNAs are the master regulators of gene expression. There are approximately 21 nucleotide small non-coding RNAs that are expressed in virtually all tissues, and changes in microRNA expression can be correlated with gene expression changes in development, differentiation, signal transduction, infection, aging, and disease. And specifically with human cancers, microRNAs are associated with cell proliferation, resistance to apoptosis, invasiveness, and differentiation in cancer cells. So a question you have is, how do microRNAs interact with mRNAs and exert their biological function? And here we provide the basis of the microRNA-mRNA interaction. This mainly revolves around the seed region, which is nucleotide positions 2 through 7 in the 5' region of your microRNA. This is the most evolutionarily conserved microRNA region. It is most frequently complementary to target 3' UTRs, and often it is sufficient to confer mRNA recognition. On the right, we have a variety of seed region matches. The basic interaction is again nucleotide positions 2 through 6. A stronger match is the 6 nucleotide match augmented by an A in position 1. Stronger yet is the 6 nucleotide match augmented by an additional base in position 8. And the strongest match is the 6 nucleotide match augmented by an A in position 1 and an additional base in position 8. Beyond the seed region, the 3' end of the microRNA can also contribute, although extensive pairing is rare. And in some cases, the central 11 to 12 continuous base pairs drives interaction. What's the result of the interaction? Nearly all cases are that you have suppression of gene expression, although there are rare cases where you observe an increase in gene expression. And for your reference, I've included a few publications at the bottom here. How do you determine microRNA-mRNA interactions? Step 1 would be to use prediction algorithms. Target prediction is based on both bioinformatics and wet lab research. For bioinformatics, the seed region match, position in the 3' UTR, cross-species conservation, as well as central sequence homology is taken into account. This is then tuned with wet lab research, including evidence from microarrays as well as reporter systems. Now, there are some pitfalls of using prediction algorithms, but that sort of goes without saying when anything that has prediction is discussed. So you have a large number of candidate mRNAs for a given microRNA, and you may not incorporate all microRNA targeting possibilities. As you can guess, different algorithms produce different target lists, and you also have the potential for the false positive rate of prediction. Now, don't let these scare you away. This is a nice first pass-through in determining what are the most relevant microRNAs for your particular sample type. You can always narrow down the list of whether a microRNA is expressed or not, or whether a target is expressed or not. That's one of the most easiest ways to narrow your list. And on the bottom, I have a few prediction algorithms that you may want to look in further. Step 2. How do you determine microRNA-mRNA interactions? You want to follow up your predictions with some experimental techniques. If you're involved with microRNA target screening, you can identify inferred targets using gene expression analysis, either through RNA-seq, microarrays, or qPCR. If you want to determine direct targets, you can use an immunoprecipitation technique, such as HITS-clip, PAR-clip, or biotin-tagged microRNA. For gene-specific validation, you can use qPCR, luciferase reporter assays, Western blots, or 5' rapid identification of cDNA ends. So naturally, when you discuss microRNA-mRNA targeting and the specific role of a microRNA, naturally, one of the next points is to discuss the role of microRNA in human diseases. So how are microRNAs involved in disease? And basically, it's any event that disrupts normal microRNA activity, which involves disruption of the microRNA-mRNA interaction. You can have alterations in transcription through promoter methylation, histone modifications, or changes in transcription factors, genomic instability events such as amplification or deletions, translocation, or insertional mutagenesis. Also, changes in either Drosha processing of the primary microRNA to a precursor, or changes in dicer processing when the precursor is processed to the mature, or ultimately loss of your microRNA binding site in your target. This is through either a SNP or a mutation, alternative splicing, or loss or change of a 3' UTR. Collectively, any of these events can disrupt normal microRNA activity. A few unique signatures in human cancers over time. microRNAs located in genomic regions amplified in cancers can function as oncogenes, whereas microRNAs located in portions of chromosomes deleted in cancers can function as tumor suppressors. Also, the abnormal expression of microRNAs has been found in both solid and hematopoietic tumors, and microRNA expression fingerprints have been correlated with both clinical and biological characteristics of tumors. And I started out today's talk with this, but overall, a substantial number of studies and reviews have associated the presence of various microRNAs with cell proliferation, resistance to apoptosis, invasiveness, as well as differentiation in cancer cells. So now let's shift gears here from a background and spend our time discussing the tools and technologies that you will use. At QIAGEN, we have microRNA quantification solutions from any sample type, ranging from non-limiting samples to extremely limiting samples. So your non-limiting samples would be your cell lines, your fresh or frozen tissue, or generally speaking, your serum or plasma when you have at least 200 microliters to process. Your typical total RNA amounts are greater than 100 nanograms. With this, you don't need preamplification. You simply synthesize cDNA and run your either primer assays or pre-formatted arrays for microRNA quantification. So limiting samples, these would be your FFPE samples, less than 100 microliters of serum or plasma, or all other fluids. For this, you would combine reverse transcription with one of our preamplification kits ahead of your either assays or arrays. And last, this is a new product we recently released. This would be your extremely limiting. This would be your individual cells or minute amounts of RNA. So ultimately, when you're working with individual cells, you will use a direct lysis method. But you can also use the single cell approach when you have very small, extremely limiting amounts of total RNA. So you would synthesize cDNA, perform preamplification, and then perform your assays and arrays. So here at QIAGEN, beyond providing you with individual solutions for sample prep, real-time PCR, etc. We have sample-to-insight solutions that can take you from your specific sample to actionable insights based off of interpretation of your data. So we have a whole host of sample prep technologies to meet your specific needs of your samples, real-time PCR assays to quantify any microRNA, data analysis solutions that are free, and data interpretation solutions to enable you to accelerate your research. So naturally, we'll begin with sample prep and discuss the offerings that QIAGEN has. So for sample prep, we offer a total RNA solution for every sample type. For cells, fresh tissue, and frozen tissue, we have the miRNA kits, the mini kit, the micro kit, and the 96 kit. The micro kit has a smaller filter size, which enables you to isolate RNA from smaller samples. And the 96 kit is actually in the 96 well plate format that enables you to process 96 samples at one time. For FFPE, we have the miRNA FFPE kit. For fluids, such as serum, plasma, urine, cerebrospinal fluid, saliva, bronchial lavage, even cell culture media, we offer the miRNA serum plasma kit. For exosome enrichment and isolation from serum or plasma, we offer the exoRNA kits. And I've also included this here that if you're interested in intact exosome purification, we offer the ExoEasy Maxi kit. And basically, the difference between the ExoEasy and the ExoRNA is that the ExoRNA includes both isolation of exosomes followed by total RNA liberation, while the ExoEasy kit simply enriches for intact exosomes. So that's it for sample prep. Let's spend now our next portion discussing real-time PCR of any microRNA. In miScript at QIAGEN is a complete microRNA quantification system. There's a reverse transcription kit, preamplification solutions for limiting RNA amounts. And again, if you're interested in single-cell microRNA quantification, we have a whole suite of products for that. For high-throughput expression analysis, we offer our high-throughput arrays. And for low-throughput microRNA quantification, we offer individual microRNA primer assays. We also offer real-time PCR reagents, a general cyber green kit, and also a microfluidics kit for the Fluidigm biomark. And this is an evergreen-based PCR master mix. Here we have the basic workflow of the miScript system. First, you isolate total RNA. Then you perform reverse transcription. Step 3, you can perform PreAMP if your samples demand it. Step 4, you prepare your PCR premix. Step 5, you load your PCR arrays or combine the mix with your individual assays. Step 6, you perform real-time PCR. And step 7, you analyze your data. And for preamplification, we'll spend a little more time just discussing a few options of miScript. And with this preamplification, you can perform micronome profiling from as little as 1 nanogram of total RNA or biofluids. This is a highly multiplexed PCR-based preamp. It is compatible with all miScript arrays and primer assays. And it enables microRNA profiling from very limited amounts of starting material. Cells or tissues, you only need 1 nanogram of total RNA. And for fluids, we would recommend preamplification when working with 50 microliters or less of serum or plasma, and any amount of basically any other fluid that you're thinking of. And when in doubt, miScript preamp it, quote unquote. Now also, we have the miScript single cell qPCR kit. And basically here I want to provide you with a product overview. And if you want any more material on the miScript single cell qPCR kit, just let me know in the question box, and I can send you a separate webinar deck discussing the whole entire single cell kit. So what is the product? It's basically lysis reagents, cDNA synthesis, and preamplification that enables universal mature microRNA quantification from individual cells. For starting materials, we recommend starting with 1 to 100 cells, or 25 picograms to 1 nanogram of purified RNA. And this would be generally considered a single-cell RNA amount and can go down to 5 to 10 picograms depending on your particular cell type. Should animal or plant samples be used? For right now, just use animal samples. But if you have plant questions, email me directly. And this is one of the times that I have my email here. I also provide it again at the end of the webinar, and it's also in the slide deck that you can download from the handout section. What products are compatible with the single-cell kit? And it's basically all of our assays and arrays, as well as our cybergreen and microfluidics PCR reagents. And what real-time PCRs instruments are compatible with the kit? It's basically all commercially available 96 and 3D4-well instruments, QIAGEN's Rotor-Gene Q, and the Fluidigm Biomark HD. For high-throughput expression analysis, I mentioned that we have PCR arrays. PCR arrays are wet lab verified primer assays that are pre-dried in PCR plates. For micronome arrays, we offer the broadest species and the broadest content available on the market. For a human alone, we offer more than 2,400 primer assays. And keep this in mind when you're also performing next-generation sequencing of small RNAs, simply because your small RNA sequencing experiments are typically followed up with a downstream validation involving qPCR, and we have the broadest assay coverage to facilitate this in your own lab. We also use our extensive bioinformatic capabilities to break the micronome into a variety of panels. First off, we break them into targeted micronome arrays, and basically these are 372 assay panels of microRNAs. We also break the micronome into biologically focused pathways and diseases. So if you're interested in a specific cancer, such as a breast or brain cancer, or a specific process such as cell development and differentiation, or apoptosis, or even a focused serum and plasma array, we have content for you. For formats, we have the 96-well, 3D4-well, and Fluidigm Biomark compatibility. And as a result of this, we have compatibility with all mainstream instruments. Importantly, this is also fully customizable. So again, I'll hit on the point of when you're following up small RNA sequencing experiments, you can easily customize a PCR array to focus only on the assays of interest. The arrays are very nice and easy to use. You simply prep your PCR reaction mix, load your plate, and run your experiment. There's no pipetting of individual primers. Here's what a PCR array looks like. We just have the 96-well as an example, but we also have 384-well. The first 84 wells contain mature microRNAs, and the final 12 wells contain a variety of controls. There's C. elegans miR39 assays that enable targeting of a spike-in that is added during fluid total RNA preparations. We also have miScript PCR controls, which are small nuclear and small nucleolar RNAs that are highly useful for data normalization when working with cells or tissues. We have microRNA reverse transcription controls and also positive PCR controls, or PPC. And what these allow you to do is monitor for reverse transcription performance and also to enable assessment of PCR performance. So we often get asked, should you use PCR arrays or individual miScript primer assays? And basically it comes down to what's best for your experiment in terms of both sample and assays. For micronome screening, we recommend always using PCR arrays, and if you're working with more than 24 assays, always use PCR arrays. Now, if you're less than 24 assays, it depends on your number of samples. So if you have a limiting number of samples, so 16 or less, simply use your individual primer assays. However, if you have an extensive number of samples, 16, 50, 100, even into the thousands as biomarker experiments often push, we recommend PCR arrays. We recommend PCR arrays because it reduces your chance for error by eliminating the need to pipette individual primers. Some performance data. The miScript system offers exceptional linearity and specificity. The reactions are linear over 6 logs of input RNA, ranging from 1 microgram to only 10 picograms. And whether your target's very robustly expressed at more than 1 million copies or very rarely expressed at only 10 copies, the system can efficiently detect it. The miScript system also offers exceptional specificity, enabling discrimination between closely related microRNA family members. In this experiment, we synthesized cDNA for each of the lead family members, and in real-time PCR used a panel of primers for each of the lead family members across the cDNA syntheses. The perfect match, primer versus cDNA, was set at 100%, and the relative detection is shown for each of the mismatches. And as you can see, the cross-reactivity is very low or non-existent. And what this shows is that even with amongst microRNAs that only vary by one base, the miScript system can efficiently discriminate. One piece of data on the single-cell kit. This kit enables detection of very low copy numbers. So for this, we have a copy number assessment of targets that were present at 10,000 copies in the original sample, or only down to 10 copies in the original sample. And this is a summary across 24 assays. And you have robust detection down to only 10 copies of a target in the original sample. This is very, very strong detection capabilities. So now let's talk about a few application examples. First off, we'll take a look at a focused microRNA expression profiling experiment. For this, we used FFPE samples. The RNA was isolated with the miRNAs FFPE kit. cDNA was synthesized with the miScript 2 RT kit. And for qPCR, we used the human miFinder PCR array. The miFinder is a great starting point. It collects the 84 best expressed, best characterized microRNAs into one plate. And here we have the raw data, 3 replicates of normal lung and 3 replicates of lung tumor. And you can see that even without normalization, the CT values are quite on top of each other. When you compare the normal versus the tumor, significant differences exist. Here we have a scatter plot of the levels of the normal lung versus the lung tumor, and anything off the axis and above or below the fold cutoff lines are differentially expressed. So you see 4 microRNAs that are substantially increased in expression and 1 microRNA that is substantially decreased. Here we have a high content microRNA expression profiling. So this would be a quote-unquote sub-micronome. For the experiment, we started with a 200 microliter serum sample, isolated the total RNA, synthesized cDNA, and used the serum and plasma 384HC PCR array. The serum and plasma 384HC collects the 372 microRNAs that we've observed the best expression for into one panel. And recently, Victor Ambrose and colleagues used the serum and plasma array to identify a biomarker signature for acetaminophen toxicity. And when you look at your normal levels versus your lung cancer serum levels of microRNA, significant differences exist, with a host of microRNAs being increased in expression. Another example, here we have HCT116 colorectal cancer cells that have been treated with the DNA methyl with 5-aza-2-deoxycytidine, which irreversibly inhibits DNA methyltransferase-driven DNA methylation reactions. And when you look at a scatter plot of levels, 104 microRNAs were strongly upregulated in the treated cells, while 30 microRNAs were strongly downregulated. And on the right, we have a volcano plot where we plot fold regulation versus p-value. And when you apply a p-value of 0.05, 89 of the 104 were significantly increased, and 21 of 30 were significantly decreased. And the nice thing is that using our free data analysis software, that all of these plots, including the scatter plot, volcano plot, clustergram, are generated automatically. Here we have a preamplification example where you can achieve excellent results with a fraction of the input. And here we had a limiting serum sample. So for the sample, we started with only 5 microliters of serum, isolated our RNA with the miRNAs serum plasma kit, synthesized cDNA, and performed PreAMP before using the serum and plasma 384HC array. As far as reproducibility, the method's very reproducible. we have an individual RNA isolation versus the CT mean of 3 isolations, and the CT values fall on a straight line with an R-squared greater than 0.97. And on the right, we have our scatter plot where we compare normal serum levels to colorectal cancer serum levels, and we're able to see microRNAs that are mildly increased in expression as well as decreased in expression. Now, the last piece of data I want to show you involves our single-cell kit. And the single-cell kit whole goal is to decipher the intrinsic transcriptional variability that is observed amongst individual cells. And often this is lost when you look at bulk analyses. And this is the whole crux of actually performing single-cell analyses, that you can remove the cellular averages that often mask your transcriptional variability. So for this, we turned again to HCT cells that were treated with 5-azo-2-deoxycytidine. And on the left, we have a bulk sample analysis. So you're able to see microRNAs that are increased in expression, as well as a few that are decreased in expression. So you do observe differential expression. However, on the right, what we have done is that we have performed individual cell analysis with the miScript single-cell kit, and perform then a principal component analysis of individual treated cells. And as you can see, you have clustering of individual cells, meaning that there's a differential response amongst treated cells with 3 distinct populations. And what this suggests is that cellular averages can often mask intrinsic transcriptional variability across individual cell populations. So now let's shift gears here from discussing sample prep and real-time PCR to discussing how to analyze your data. In real-time PCR data analysis, while there are a variety of methods, 2 of the main ones are absolute quantification, which involves calculating an absolute input copy based on a standard curve, and relative quantification, which is also known as the comparative CT method, or 2 to the negative delta delta CT method. This involves selection of an internal control and also the selection of a calibrator, for an example, an untreated control or a normal sample. And again, for your reference, I've included a few publications. So your data analysis workflow for relative quantification is as follows. You perform your real-time PCR experiment and set your baseline and threshold to determine your CT values. Step 3, you export your CT values. And step 4, you analyze your data using the delta-delta CT method of relative quantification. So let's go through the steps. Data analysis step 1 is setting your baseline. Your baseline is defined as your noise levels in your early cycles, where there's no detectable increase in fluorescence due to PCR products. How should you set your baseline? You should observe your amplification plot using linear view and determine your earliest visible amplification. From there, you set your baseline from cycles 2 or 3 to 2 cycles before your earliest visible amplification. And as a general note, your number of cycles used to calculate your baseline can be changed and should be reduced if high template amounts are used. And very importantly, be sure that your baseline settings are the same across all PCR runs. Your threshold, the purpose is that it is used to determine the CT or threshold cycle value. And the point at which the amplification curve intersects with the threshold line is called the CT. How should you set this? First off, observe your amplification plots using log view, and you place your threshold in the lower half of the log linear range above the background signal. As a first note, be sure to never set your threshold in the plateau phase. And like the baseline, be sure that your threshold settings are the same across runs being analyzed. So step 3, you export your CT values. And as the example here, we'll just use our FFP experiment we talked about earlier as a frame of reference. So for the experiment, you would have performed 3 total RNA isolations from normal lung and 3 from lung tumor, synthesized cDNA for each, performed real-time PCR, and exported your CT values. Now the next step is that you need to normalize your CT values for each microRNA. This involves a calculation of the CT of your microRNA minus the CT of your normalizer. And for this case, we used an FFPE sample, so it's acceptable to use small nuclear, small nucleolar controls. And often it's very perfectly acceptable to use an average of multiple controls. And as a tip for choosing an appropriate control, be sure that your selected controls are not influenced by your experimental condition. So once you've calculated your delta CT, i.e. normalized your CT values, you then calculate the average delta CT for each microRNA within your group. This involves adding the delta CTs and dividing by your number of replicates. From there, you calculate the delta-delta CT for each microRNA between each group and its test versus reference. So in our case, it's delta-delta CT equal to the average of the delta CT of your tumor minus the average delta CT of your normal. From there, you linearize and calculate your fold change for each microRNA, tumor versus normal, using the equation 2 is to negative delta-delta CT. So looking at a couple examples of how to calculate this, first off, we'll look at an increased expression of a target in a tumor sample. Two conditions, normal and tumor. For the microRNA, we'll look at, as an example, miR21 that has a CT of 21 in normal and a CT of 15 in our mock tumor sample. For a normalizer, we'll use RNU6 that has a CT of 16 in normal and CT of 14 in tumor. Step 1, normalize your data. So this is CT of your microRNA minus CT of your normalizer for both normal and tumor. Step 2, calculate your delta-delta CT, tumor relative to normal. So this is then delta CT of tumor, which is 1, minus delta CT of normal, which is 5, to give you negative 4. Step 3, calculate fold change using the equation 2 is to negative delta-delta CT. So this is then 2 is to negative 4 to give 16. From there, calculate fold regulation. If the fold change is greater than 1, the fold change results may then be reported as a fold upregulation. And stated then, compared to the normal sample, miR21-5P is 16-fold upregulated in the tumor sample. Data analysis example 2, we'll take a look at a decreased expression in the tumor sample. For example, miR16, CT of 15 in normal and CT of 16 in tumor. First off, normalize your values. First, for normal, we have 15 minus 16 to give negative 1, and 16 minus 14 to give 2 for the tumor sample. Then you calculate delta-delta CT, 2 minus negative 1 to give 3. Then you calculate fold change using the equation 2 is to negative delta-delta CT. So it's 2 to the negative 3 to give 0.125. For calculation of fold regulation, if the fold change is less than 1, which is our case, the negative inverse of the result may be reported as a fold downregulation. So this is negative 1 divided by 0.125 to give negative 8. Stated then, compared to the normal sample, miR16-5P is 8-fold downregulated in the tumor sample. So serum and plasma is a special data analysis case, and we'll spend a few slides discussing this. So serum and plasma samples are special because the SNO-RNA, snRNA panel of targets does not exhibit robust expression and should not be selected as normalization controls. For example, here are typical CT values that you observe, and in serum and plasma samples, the CT values are often in the 30s. Often they're not even detectable at all, so those are two very good reasons to not use small nuclear and small nucleolar controls for serum and plasma data normalization. So instead, your options are as follows. You can calibrate your samples using a C. elegans miR39 mean, and then normalize your serum or plasma to either all commonly expressed microRNAs or to invariant microRNAs. And we'll spend the next couple slides going over this. So first off, calibration using the C. elegans miR39. You can only do this if you've spiked in the C. elegans mimic or the microRNAs serum plasma spike-in control during biofluid sample preparation. And basically, the assay targets the C. elegans and can account for any differences due to differences during the total RNA isolation procedure. So for example, if you look at an uncalibrated sample, sample 2 compared to sample 1 appears to have a delayed CT value. 3 units for miR16, 4 for 21, 3 for 192, and 3 for 25. At the same time, you also see a 3 CT unit delay for the C. elegans. What this suggests is that this indicates a differential recovery. So as a result, you would calibrate your samples, and this involves calculating a correction factor, which you take all CT values in sample 2 and subtract 3, based off your difference of the C. elegans. And once you account for differential recovery, you can see that there's no delay for miR16, only a 1 unit delay for miR21, and the CT values for miR103 are actually 3 units more robust in sample 2. So then once you've calibrated your data, you have 2 normalization options. Option 1 is to normalize to the CT mean of expressed microRNAs. And what this involves is calculating the CT mean for commonly expressed microRNAs. And those are microRNAs with a CT value less than 30, or 32, or 35. It depends on your limit of detection in all of your assessed samples. Option 2 is to normalize to the CT mean of invariant microRNAs. And this involves identifying at least 4 to 6 microRNAs that exhibit little CT variation and using the CT mean for normalization. On the left here, we have a panel of commonly expressed microRNAs in serum and plasma. And basically, I'm not saying that these are invariantly expressed. It just means that we typically observe expression for these in serum and plasma samples. So when you compare the normalization methods, you would use the commonly expressed microRNA method when working with micronome profiling or one of our PCR arrays. You would choose invariant microRNA normalization when working with a small panel or individual assays. In this example, as a first note, the fold regulations look highly similar between the 2 methods, and this is correct as your results should be as independent of your chosen normalization method as possible. And also, as a second note, for small panel screening, do not use a CT mean of all microRNAs as this array is biased, meaning that your assays are not included on this array at random. So now for using the GeneGlobe Data Analysis Center, what this tool enables you to do is perform delta-delta CT calculations with a few clicks of your mouse. Steps 1 through 3 remain the same. You set your baseline and threshold, and then export your CT values. But step 4, you access the free data analysis software at qiagen.com/GeneGlobe and perform automatic data analysis using the delta-delta CT method. For the data analysis software, this is a web-based software where no installation is needed and is tailored for each individual array. Basically, you can go from raw CT values to results using the delta-delta CT method. And as I had previously mentioned, your data is represented in multiple visual outputs, including a scatter plot, volcano plot, multi-group plot, and clustergram. So last here, we'll discuss data interpretation. So first off, the Ingenuity pathway analysis available through QIAGEN asks what's next in your data by modeling, analyzing, and understanding complex omics data. This enables the analysis of gene expression, microRNA, SNP, and SNP microarray data, as well as qPCR and next-generation sequencing data as well. And what it enables is a deeper understanding of metabolomics, proteomics, and RNA-seq data. And what I'd like to describe it as a very intelligent PubMed. So instead of just having raw publications, what Ingenuity does is it establishes connections between particular publications So instead of just getting a list, you can understand the relationship of what publications are actually connected to each other. So there are more than now 5 million connections in the Ingenuity database. And what this can do is very quickly accelerate your research, enabling you to identify if your hypothesis has been successfully tested. Other things of data interpretation involve manipulation of microRNA function. And actually, we have a webinar next week regarding manipulating microRNA function through mimics and inhibitors. Mimics mock endogenous microRNA activity and act just like a natural microRNA. And microRNA inhibitors block microRNA activity and prevent a microRNA from interacting with and downregulating its intended mRNA target. All right, so let's finish up here. Where can I find the products discussed today? These would be at QIAGEN.com or QIAGEN.com/GeneGlobe. GeneGlobe is a very nice one-stop shop for all things biology at QIAGEN. When you come to the homepage for GeneGlobe, you see the search box. You can search for your gene expression target of interest, a microRNA of interest, a biological pathway or disease of interest, and you receive a whole host of products that are associated with that search. I have my agenda slide, one out of order. But here we have the conclusions for today. So what we have covered are that microRNAs are the master regulators of gene expression, and QIAGEN has a sample to insight solution specifically tailored for you with respect to sample prep, real-time PCR assays, data analysis, and data interpretation. And collectively choose QIAGEN to turn your hypotheses into actionable insights. So with that, we'll take any questions. I'd like to thank you for attending today's webinar. Feel free to write down my email, jonathan.schaefer@qiagen.com, and also the general QIAGEN contact information at 1-800-426-8157 or brcsupport@qiagen.com. So with that, let me take a look at the question.